Welcome to Bookfulness. This is episode 20.
And we are wrapping up our fourth major unit in this deep dive. Just a quick reminder, last time we were looking at the dynamics of diversity. How species pile up, how they interact, and eventually extinction.
Right. Today we're doing the opposite, really. We're pulling way back, zooming out to the widest possible lens, the really vast ancient sweep of history.
Yeah.
We're talking macroevolution, so evolution above the species level. And our guide here is Chapter 20 of Evolution, 4th edition.
Yeah, and our mission today basically is to connect the dots. How do those small, generation-to-generation changes, what we call microevolution, how do they add up over immense geological time? How do they produce these grand, large-scale patterns and, major new forms of life like vertebrates or flight?
So are we talking about fundamentally different kinds of evolution, or is it just more the same but over millions of years?
That's the core question, really. Is macroevolution something special, needing unique processes, maybe huge genetic leaps? Or is it just the combined power of things like selection and drift, playing out incredibly slowly, accumulating over all that deep time?
Okay, let's unpack that. Because when we say macroevolution, we're talking big picture stuff.
Yeah.
The origin of whole new classes, orders, families, entirely new biological structures.
Exactly. It's the difference between, say, a finch evolving a slightly different beak shape, that's micro, and the whole massive transition from, a land-dwelling reptile ancestor to a fully feathered flying dinosaur or bird. That's macro.
Okay, so let's start by really nailing down that definition. Macroevolution. It's evolution above the species level. Simple as that, definition-wise. It covers processes unfolding over geological time, shaping how whole groups, clades, diversify or go extinct.
And you absolutely need to contrast it with microevolution, the small-scale stuff happening within populations. Natural selection, genetic drift, mutation, gene flow, those four familiar forces.
Right. And historically, before we had the modern evolutionary synthesis, the blending of genetics and Darwinian selection, people did think macroevolution needed its own special rulebook.
Oh, definitely. Some very smart people proposed unique, powerful mechanisms, something bigger than just, like, moths changing color near factories. They thought originating a whole new body plan required something different.
But the modern view really solidified around the mid-20th century, thanks to people like the paleontologist George Gaylord Simpson. That view is different.
Completely. The consensus now is that those big patterns we see in fossils, they're built entirely from those familiar microevolutionary processes. Macroevolution isn't some separate force. It's just microevolution with a much, much longer timeline.
So the difference is about scale,
Yeah.
about time, quantity, not quality.
Precisely. And to really get a feel for that scale and how small changes can add up, we need a concrete example. A major transition, and, probably the best documented one, the most complex, is the origin of mammals.
From their reptile-like ancestors, the synapsids. Specifically, a group called the cynodonts.
That's the one. Think about an ancestral cynodont, maybe 250 million years ago. Compare it to a modern mammal. The differences are huge. Hair, warm-bloodedness, milk production, that incredible middle ear.
Did all that just pop up at once, a sort of mammal package deal?
No way. And that's maybe the most important concept here, mosaic evolution. It's the rule for these big transitions. Different key mammalian traits evolved at different rates at different times. It wasn't simultaneous.
Like assembling a toolkit, piece by piece, over millions of years.
Exactly. The defining mammalian characteristics were put together gradually.
Okay, let's break down that mosaic. Maybe start with something basic like teeth. How did feeding change? Ancestral cynodonts, like a lot of reptiles, had pretty simple teeth, right? All basically the same shape.
That's right. Homodont teeth, fairly uniform. But look at a mammal today. Incredible specialization. We've got sharp incisors for cutting, long canines for grabbing, complex molars and premolars for grinding, shearing.
All sorts of different tools in the mouth.
Which lets mammals process food way more efficiently.
Yeah.
That generates more energy, which you need for things like being warm-blooded. And this dental differentiation, it was actually one of the earlier pieces of the mammalian toolkit to start evolving.
Interesting. But the really mind-bending change, the one that makes you just marvel at how evolution tinkers, has got to be the jaw joint and how that relates to the middle ear.
Oh, absolutely. It's a brilliant, brilliant example of evolutionary remodeling. We should walk through the anatomy because it's stunning.
Okay, lay it out. The ancestral setup in those cynodonts, the synapsids.
So their jaw hinge used the standard reptilian connection. A bone in the skull called the quadrate, let's call it the Q bone, connected with the bone in the lower jaw called the A bone. So a cubed joint. Functional, sure, but not great for really complex chewing.
And in the line leading to mammals, what happened? What took over?
Well, the lower jaw in synapsids was actually made of several bones. But over time, one of those bones, the dentary, the dead bone, which makes up pretty much our entire lower jaw, now it got bigger, much bigger and stronger. Okay. And the functional connection, the hinge point, it started to shift. It moved to where this bigger dentary bone connected with a bone in the skull called the squamosal, the Sioux bone. This new DSQ joint became the main jaw joint in mammals.
Why the change? What was the advantage?
A much stronger bite. This new joint allowed for way more muscle attachment, much better mechanical strength. Absolutely crucial for the specialized chewing that mammals do.
Okay, wait. So are you saying they kind of evolved a new jaw joint while the old one was still hanging around? That sounds a bit awkward.
Well, yeah, that's the nature of these transitions. For a while, in those intermediate forms, they likely had a double jaw joint. Both the old QA and the new DSQ joints might have been functional at the same time.
Huh. Like having a backup system while the new one gets road tested?
Exactly. It provides redundancy during a massive functional shift. You can't just switch off the old system instantly.
Makes sense. But then, the big question. What happened to those original jaw joint bones, the articular and the quadrate? Once the dentary and squamosal took over the main jaw, did they just disappear?
This is the truly astonishing part. They didn't disappear. They were repurposed, exquisitely co-opted for a completely different job, a sensory job.
Okay.
The articular bone, the A bone from the old lower jaw joint, it was modified, shrunk down, and became the malleus, or the hammer bone, in the mammalian middle ear.
No way.
Yes way. And the quadrate bone, the Q bone from the old skull joint, it became the incus, or the anvil bone, two of the three tiny little bones, the ossicles, that amplify sound vibrations in our middle ear.
That is genuinely incredible. Bones that were part of the jaw for chewing get detached, miniaturize, and become part of the hearing system.
Right. The third ear bone, the stapes, was already there in ancestral tetrapods doing a basic sound transmission job. But adding the repurposed malleus and incapacious, that dramatically improved hearing sensitivity, especially for higher frequencies.
What would be the advantage there? Why invest in better hearing?
Think about those early mammals. Many were probably small, maybe nocturnal, trying to survive in a world with lots of larger reptile-like predators. Being able to hear really well, especially subtle, high-pitched sounds in the dark, that would be a huge survival advantage.
So they got a stronger bite and better hearing, all by remodeling existing parts.
That's mosaic evolution and co-option in action, incremental modification and repurposing.
And the amazing thing is, this isn't just guesswork. We actually have fossils that show these intermediate stages, right?
We absolutely do. Fossils like Snauconodon from the early Jurassic, it shows a mix of features. And then there's the tiny shrew-sized hadrocodium. It's one of the earliest critters we'd confidently call a mammal, and it already has that derived middle ear structure, even though it was minuscule.
So the fossil sequence backs up this gradual, step-by-step mosaic assembly over millions of years.
Concrete evidence. It really drives home that these major transitions are built from gradual changes.
And just a note for you listening, if you want to really visualize this jaw-to-ear transition, because it is pretty complex but also elegant, we've put links in the description to the key figures from the textbook, figures 20.1 and 20.2. They really help make it clear. It wasn't a sudden leap. It was incredible anatomical remodeling.
So that mammal transition is a fantastic case study for Darwinian gradualism change happening through many small successive steps. But, throughout the history of evolutionary biology, people have pushed back against that idea. They've often favored the notion of big evolutionary leaps or saltations.
Right. This is where we get into things like the hopeful monster idea, which always sounds so dramatic.
It does. The geneticist Richard Goldschmidt, back in the 1930s, 1940s, he was really bothered by what seemed like big gaps in the fossil record. He argued that simple, gradual, Darwinian change couldn't explain the origin of major new groups, what he called higher taxa.
So what was his alternative?
He proposed systemic mutations, basically huge simultaneous genetic changes that would create a radically different organism in just one generation. If, by some miracle, one of these massively different mutants actually survived and could reproduce, it would be a hopeful monster, potentially founding a whole new lineage adapted to a new way of life.
I mean, I can sort of see the appeal if you're just looking at fossils with big gaps between them. It looks like a jump. But surely, from what we know about genetics now, that kind of massive coordinated change is just impossible.
Yeah, it's been completely refuted. Modern genetics and developmental biology show us just how interconnected everything is in an organism. A huge, simultaneous genetic overhaul, the kind Goldschmidt envisioned. It's almost guaranteed to be lethal, or at the very least, severely disadvantageous.
Would mess up too many things at once.
Precisely. Development is a finely tuned process. Making massive, coordinated changes successfully in one go is overwhelmingly unlikely. Evolution has to build on what's already there, tweaking and modifying functional systems, not trying to invent entirely new ones from scratch in a single generation.
Okay, but let's be fair. Even today, we look at some groups, and the gaps do seem enormous. Like the difference between any land mammal and a whale or a dolphin. There are no living intermediates bridging that huge morphological gap.
That's a classic example, and this is where we really need to separate an actual biological leap from an apparent gap in the record. Those conspicuous gaps, they're usually just artifacts
Artifacts of what?
of the fossil record being incomplete. Fossilization is a rare event. Lots of intermediate forms simply didn't get preserved or haven't been found yet. And crucially, extinction prunes the tree of life. When those intermediate branches die out, the surviving endpoints look much farther apart than they actually were, evolutionarily speaking.
And the whale story is actually a fantastic counter-argument against saltation, isn't it? Because we have found intermediates now.
Exactly. Go back a few decades, before the 1980s or 90s, and the whale transition looked incredibly abrupt. Then came discoverers in places like Pakistan. Fossils started filling in the gaps beautifully.
Like what?
Fossils, like Pakicetus, looked a bit like a furry wolf, but its ear structure showed clear whale affinities and it lived near water. Then Ambulocetus, the walking whale, it had large hind limbs but could clearly swim effectively. We see nostrils migrating backwards up the skull over time, hind limbs shrinking.
Step-by-step adaptations to aquatic life.
Precisely. Figure 20.3 in the text shows some of these key fossils. Extinction just removed those stepping stones from the modern world, leaving the highly specialized whales and dolphins looking isolated from their closest living relatives, which are actually hippos.
Okay, so gradualism holds for mammals and whales.
Yeah.
What about something that seems truly uniquely bizarre, like the turtle shell? That structure is unlike anything else among amniotes. How could that evolve step by step?
The turtle shell. It's maybe the best example we have of a major complex novelty arising from a subtle but profound shift in development.
What's the key anatomical weirdness?
It's the relationship between the ribs and the shoulder blade, the scapula. In all other amniotes, birds, lizards, crocs, mammals, our ribs develop underneath the shoulder girdle. But in turtles, the ribs grow over the scapula.
Which is structurally necessary for them to fuse together and form that protective dome, the carapace.
Right. And that fundamental rerouting of the ribs seems to be the key innovation. It wasn't about evolving the shell bones first, but about changing the developmental pathway of the ribs.
How did that happen?
It involves the evolution of a new embryonic structure, something called the carapacial ridge, or CR. It appears along the flank during development, and crucially, this ridge sends out signals to the developing ribs nearby.
And what do those signals do?
They basically tell the ribs, stop growing down into the body wall like you normally would. Instead, they redirect the ribs to grow outwards and upwards over the pectoral girdle.
So the real novelty wasn't the shell itself initially, but the developmental instruction, the signal that changed where the ribs went.
That seems to be the case. And we have a fossil that supports this idea beautifully. Odontochelys semitestacea. It's a Triassic turtle, one of the earliest known.
What did it look like?
It's fascinating. It didn't have a fully formed carapace, the upper shell, on its back, but it did already have the plastron, the belly shell, and critically, its ribs, especially the front ones, were already broad and flat and clearly starting to grow outwards and redirect towards the back, over the shoulder girdle.
So the rib redirection happened before the full protective shell was complete.
Exactly. Figure 20.4 shows this fossil. It suggests the underlying developmental change came first, setting the stage for the later evolution of the full carapace. It's a gradual buildup, starting with a change in developmental signals.
This also touches on something we mentioned earlier. Sometimes a big-looking change, a big phenotypic change, can happen relatively quickly, but the genetics behind it might be quite simple. Not quite Goldschmidt's idea, but still rapid.
Yes, that's a really important distinction. You can get large phenotypic effects from relatively simple genetic changes if those changes happen in certain hotspot genes.
What makes a gene a hotspot?
These are often genes that act as key regulatory switches in development. A mutation here can have a big effect downstream, but critically, for it to work evolutionarily, the mutation shouldn't have widespread negative side effects, what we call deleterious pleiotropic effects.
Can you give an example?
The ambystoma salamanders are perfect. You've got the tiger salamander, which goes through metamorphosis, loses its gills, develops lungs, and walks on land. Then you have the axolotl, which stays in the water its whole life, keeps its gills, basically remaining in a larval form.
They look completely different as adults.
Totally different lifestyles and morphologies. But the genetic difference causing this is surprisingly small. Just a few genetic changes essentially shut down or truncate the hormonal pathway that triggers metamorphosis. It's called paedomorphosis, retaining juvenile features as an adult.
So why isn't that a hopeful monster kind of jump? The effect is huge.
Because you're not creating brand new complex systems out of thin air. You're primarily stopping or modifying an existing complex developmental process, metamorphosis. You're altering a regulatory switch, not simultaneously re-engineering multiple organ systems. The change looks big, but the underlying genetic disruption is relatively localized and manageable.
It's more like hitting a pause button on development than trying to build something entirely new overnight.
That's a good way to put it. And we see similar things in insects too, like in the fruit fly Drosophila cicalia, the loss of those little hairs, the trichomes on its back. That seemingly significant morphological change is caused by mutations in just a few of these hotspot genes. It shows rapid divergence is possible without invoking impossible genetic leaps.
Okay, so evolution builds gradually. It avoids massive disruptive leaps. It tinkers with development. But that still leaves the question, where do genuinely new things come from? How do you get something like an island or wings or a bee stinger if you're always just modifying what's already there?
This is where co-option and exaptation become absolutely central. Co-option is basically recruiting existing structures or genes or pathways for a completely new job.
Taking something that evolved for purpose A and using it for purpose B.
Exactly. And the new function itself, purpose B, is often called an exaptation. Evolution is the ultimate tinkerer, the ultimate recycler. It rarely designs things from scratch. It grabs whatever spare parts are lying around and finds a new use for them.
Okay, give us some classic examples of this evolutionary recycling.
Well, we already talked about the jaw bones becoming ear bones. That's a prime example of co-option. Another famous one is the stinger in bees and wasps.
What was that originally?
It's a modified ovipositor, the organ that their ancestors used to lay eggs. In stinging insects, it's been co-opted into a venom delivery system for defense or predation, from reproduction to weaponization.
Wow. Okay, what else?
Think about penguin wings. They are clearly homologous to the wings of flying birds, same bones, same evolutionary origin, but penguins don't fly in the air.
Right. They fly underwater.
Exactly. The wing structure, originally adapted for aerial flight, has been exapted for powerful underwater propulsion. The basic structure was there, perfectly suited for a modification into a flipper.
And this happens at the molecular level too, right? With proteins and genes?
Oh, absolutely. The proteins that make up the lens in our eyes, the crystallins, they make the lens transparent and help focus light, but they didn't evolve for sight originally.
What were they doing before?
Many of them were originally stress proteins, like heat shock proteins. Their job was to help other proteins maintain their shape and function under cellular stress. But they happened to have properties being stable, transparent, water-soluble that made them perfect building blocks for a lens. So selection recruited them, co-opted them for vision.
So a protein that helps cells cope with heat stress ends up helping us see the world. That's amazing.
It really is. Or think about waste products. Purine butterflies, like the cabbage white, they excrete nitrogenous waste as uric acid. That's just metabolic necessity. The white color is just a chemical property. But these butterflies started storing this white uric acid in their wing scales. Suddenly, this waste product became a pigment. And that pigment turned out to be useful.
How so?
It can act as camouflage, help regulate body temperature by reflecting sunlight, and even be used in signaling between males and females. A non-adaptive byproduct gets co-opted into multiple adaptive functions.
This whole idea of gradual building and repurposing seems like a direct answer to that old argument about irreducible complexity.
It absolutely is. That's the argument often used against evolution, that some structures, like the eye, are supposedly so complex that they couldn't possibly function if any single part was missing. Therefore, they must have been created all at once in their final form.
Like a mousetrap, take away one part and it doesn't work at all.
Right, but Darwin himself anticipated and refuted this even without knowing the genetics or molecular details.
How did he argue against it?
He pointed out that if you can imagine a series of small, gradual steps, each providing some advantage, however slight, then complexity can be built up over time. You don't need the final perfect version from day one. Start with just a light sensitive spot that's better than nothing. Then maybe curve it into a cup to get directionality. Add a simple lens. Each step is functional and advantageous.
And modern biology completely backs him up.
Completely. We see eyes of all intermediate grades of complexity in living animals today, showing the plausible steps. And molecular biology reveals the co-option story. The basic components needed for vision, light-detecting molecules, pigment shields, lens proteins were already present in ancient ancestors, doing other jobs.
So evolution had a pre-existing toolkit of parts.
Exactly. A toolkit that could be assembled and reassembled independently in many different lineages to build functional eyes suited to different needs. There was no need for an impossible leap.
This idea of a shared ancient toolkit, that leads us towards another really deep concept, doesn't it? Something called deep homology.
Yes. Let's clarify terms first. We know homology is similarity due to shared ancestry, like the bones in our arm and a bat's wing. Biological homology is a bit broader, referring to similarity based on a shared genetic and developmental program, maybe even for repeated parts within one organism, like vertebrae.
Okay, so what makes deep homology deep?
Deep homology refers to the reuse of very ancient conserved genetic pathways, sets of interacting genes to build structures that might look completely different, often in very distantly related animals. The genetic instructions are ancient and shared, but the final output, the structure, can be wildly divergent.
Can you give the classic example?
The PAX6 gene is the textbook case. It's a master control gene that kicks off eye development. Homologs of PAX6 do this job in animals as different as us with our camera eyes and insects like fruit flies with their compound eyes.
So the eyes themselves look totally different, evolved independently in many ways.
But the fundamental genetic switch that says build an eye here is incredibly ancient and shared across these vast evolutionary distances. That shared genetic pathway is the deep homology.
That's a really crucial point. It highlights both the constraints and the creativity of evolution.
Exactly. Evolution is constrained because it often reuses the same old genetic toolkits. It doesn't invent entirely new pathways for everything, but it's creative because it can deploy those ancient pathways in new contexts at different times and places during development to produce novel outcomes.
Any other examples of these deep pathways?
The distalis gene pathway is another great one. It seems to be involved in patterning things that grow out from the body wall, what we call evaginations.
Like what?
Like insect legs, antennae, even the weird horns you see on some beetles, or the tube feet of starfish. These structures look very different, but the underlying genetic instructions involved in their initial outgrowth share this deep homology, tracing back to a common ancestor hundreds of millions of years ago. The ancient toolkit gets used again and again, but molded into different final forms.
All right, so we've got novelty arising gradually, often by repurposing old parts, guided by these deep genetic pathways. Now let's talk about the speed of evolution, the rate of change. And this is where things get a bit weird, comparing what scientists measure today versus what the fossil record shows over long time scales.
Yeah, this is a really interesting paradox, and resolving it is key to linking micro and macroevolution. We can measure evolutionary rates in the short term in labs or field studies over a few generations. We use units called haldanes, which measure change in standard deviations per generation.
And what do those short-term measurements show?
They show incredibly fast rates.
Yeah.
Hundreds, thousands, even millions of times faster than the average rates we calculate from the fossil record over millions of years.
That seems counterintuitive. The book had that striking example at typical short-term rates. You could theoretically evolve a mouse into something the size of an elephant in, what, less than 100,000 generations?
Yeah.
Which is actually not that long, geologically speaking.
Right. So the paradox is, if evolution can happen that fast, why doesn't the world look like it's constantly and rapidly transforming? Why aren't we seeing mice turn into elephants all the time?
Okay, why not?
The key is that those high rates measured over short periods. They're simply not sustained over the long haul. They represent rapid responses to temporary or localized environmental pressures.
Like a population quickly adapting to a short-term drought or a new predator.
Exactly. But over millions of years, environments fluctuate. Selection pressures change direction. Maybe the climate oscillates back and forth, or selection favors an optimal size and keeps pulling the population back towards the average, stabilizing selection. Those rapid bursts of change in one direction often get canceled out by later changes in another direction, or just fizzle out.
So the slow rates we see in the fossil record are like the net result. The average after all the short-term ups and downs have smoothed out.
Precisely. The fossil record captures the net change over vast stretches of time. It filters out the short-term noise and reveals the cumulative directional signal, if there is one. Those high short-term rates are real, but they rarely translate into sustained long-term trends.
This realization that change isn't always slow and steady, but might involve rapid bursts followed by long periods of stability that really changed how paleontologists interpreted the fossil record, didn't it? Leading to the whole punctuated equilibria debate.
It did. Before that, the prevailing view was often phyletic gradualism, the idea that lineages change slowly, constantly, and gradually over time. But when paleontologists looked closely, they often saw something different.
Which was?
Stasis. Long periods, millions of years sometimes, where a species' morphology stays the same. Negligible change. The book gives the example of a bivalve, Macroevolution immaculata. Specimens look basically identical over a 17 million year period. Stasis seems to be incredibly common, maybe even the dominant pattern for many species.
Wow, 17 million years of just being the same. That definitely doesn't fit the slow and steady gradualism picture.
Not at all. And this observation of widespread stasis led Niles Eldridge and Stephen Jay Gould back in 1972 to propose punctuated equilibria, or PE.
Okay, break down PE. What's the core idea?
The core idea is that evolutionary history is characterized by these long periods of stasis, which are then punctuated by relatively brief bursts of rapid evolutionary change. And crucially, they argued that most of this significant change happens during the process of speciation.
Especially in small, isolated populations.
Yes, often in peripheral isolates, away from the main range of the species. The idea is that genetic drift and strong localized selection can cause rapid divergence in these small groups. If they become reproductively isolated, speciate and then perhaps expand, they appear suddenly in the fossil record alongside their relatively unchanged ancestor.
And because the change happens fast, geologically speaking, and in a small localized area, the chances of finding fossils of that actual transition are really low, which explains the apparent gaps.
That was a key part of the argument. Now, it's fair to say the fossil record strongly supports the prevalence of stasis, but the strictest version of PE, the idea that all significant phenotypic change is tightly linked only to speciation events, that's still debated. We do have examples where large, widespread populations seem to evolve gradually too.
So maybe it's not an easier situation? Stasis is common, rapid change happens, sometimes linked to speciation, sometimes not.
That's probably closer to the current consensus. But speciation still plays a really important macroevolutionary role, even if it isn't the only engine of change. It acts more like a stabilizing mechanism.
How so? What does speciation stabilize?
It stabilizes divergence. Think about it using figure 20.20 from the text. Imagine two populations of a species get separated geographically, say, by a mountain range. They start adapting to their local conditions, diverging genetically, maybe developing unique features.
Okay, they're becoming different.
But what happens if that barrier disappears? If the mountain erodes and they come back into contact?
If they haven't speciated yet, they can just interbreed, right?
Exactly. And if they can still interbreed freely, gene flow will mix everything back together. Those unique adaptations, they each evolved. They get diluted, broken down by recombination, the divergence is lost, the populations might just merge back into one.
But if reproductive isolation has evolved while they were separated, if they've become distinct species, then even if they meet again, they can't effectively interbreed.
Precisely. Speciation acts like a ratchet. It locks in the divergence. Once reproductive isolation is established, the two new species can coexist or track their preferred environments without their distinct adaptations being eroded by gene flow.
So a series of these speciation events, each locking in a bit of adaptive change that could drive a long-term trend.
Yes. It allows divergence to accumulate step by step over millions of years. Speciation captures or ratchets the adaptive changes achieved in isolation, making them permanent features of that new lineage. It provides a mechanism for converting short-term, potentially reversible local adaptation into long-term, stable macroevolutionary divergence.
Okay, so speciation can help lock in change and potentially drive these long-term directional shifts that we see in the fossil record. We call these shifts trends. But we need to be careful with terminology here, right? Trend versus progress.
Absolutely crucial distinction. A trend is just an objective, observable, directional pattern over time. The average body size in a lineage gets bigger, or the number of segments in an appendage consistently decreases. It's measurable.
Whereas progress implies something more.
Progress implies improvement, betterment, movement towards some kind of defined goal or superior state. And that requires a subjective value judgment. Who decides what's better? Evolution doesn't work towards a predetermined goal. It favors whatever works right now in a particular environment. There's no inherent ladder of progress in evolution.
Good point. Okay, so sticking to objective trends, how do these directional patterns actually arise? Are they always driven by some consistent force?
Not necessarily. We can generally think about two main models for how the average value of a trait can shift directionally over time: passive trends and driven trends. Figure 20.21 illustrates this nicely.
Let's start with the passive trend. What's that?
A passive trend can happen even if the underlying evolutionary change is essentially random, drifting in all directions equally. The key is that there's some kind of boundary or constraint that prevents movement in one direction.
Well, you can't get smaller than a certain minimum viable size.
Exactly, that's a classic example. Life has to start somewhere, presumably near a minimum level of size and complexity. If subsequent evolution involves diversification and random changes in size, lineages can get bigger, but they can't get smaller than that minimum boundary.
So even if changes are random, the average size of the whole group will tend to increase, just because the variation can only expand away from that lower limit.
Right. The variation spreads out, the maximum value increases, and the mean gets pulled along with it, away from the boundary. It looks like a directional trend, but there isn't necessarily a consistent selective force pushing everything towards being bigger. It's more about the expansion of variance away from a floor.
Okay, so that's passive. What about a driven trend or an active trend?
That's different. In a driven trend, there is a consistent bias.
Hmm.
Changes in one particular direction are consistently favored over changes in the other direction. There's a directional force, usually natural selection, pushing the lineage.
So selection consistently favors, say, larger body size generation after generation.
Yes. In that case, you'd expect the entire distribution of sizes, the minimum, the maximum, and the average, to shift upwards over time. The whole range is being actively dragged in one direction.
A famous example often discussed is Cope's rule. What's that again?
Cope's rule is the observation that many animal lineages tend to show a trend towards increased body size over evolutionary time. We see it quite clearly in some groups, like the horse lineage, for example.
So is that a passive trend or a driven trend? What causes it?
That's where it gets interesting, because a trend like Cope's rule could potentially be driven by processes acting at two different levels.
Okay, what are the levels?
First, it could be driven by simple, consistent individual selection acting within the populations or species of that lineage. This is pure microevolution. Maybe being bigger consistently gives an individual animal an advantage. Better predator defense, more efficient metabolism, better luck in competing for mates. So, within each species, selection favors larger individuals over time.
Makes sense. What's the other level?
The trend could also be driven, or at least reinforced, by species selection. This is a genuinely macroevolutionary process.
How does species selection work?
It operates on the properties of species themselves, not just individuals. The idea is that species with certain characteristics, like perhaps larger average body size, might have a higher probability of speciating, giving rise to new species, or a lower probability of going extinct compared to species with smaller body size.
So it's not necessarily that being big makes an individual horse better, but that lineages composed of larger horse species tend to last longer or branch more often over millions of years.
Exactly. Look at figure 20.22. If large-bodied species, for whatever reason, maybe they have wider geographic ranges making them less extinction-prone, tend to survive longer through geological time, or if they tend to bud off new species more frequently, then over the grand sweep of macroevolution, the whole clade will appear to trend towards larger size, even if individual selection within any given species isn't always pushing for bigness.
So the overall trend emerges from the differential birth and death rates of entire species.
Correct. It's selection acting on species as units. Disentangling whether a trend like Cope's rule is due to individual selection, species selection, or maybe a combination of both is actually a major challenge in macroevolutionary research.
Okay, let's apply that thinking to maybe the grandest trend people perceive. The trend towards increasing complexity in the history of life. We went from simple cells to multicellular organisms to complex animals with brains. Is that a driven trend? Is evolution actively selecting for complexity?
Well, people like John Maynard Smith and Ears Zathmary identified what they called the major transitions in evolution: the origin of chromosomes, eukaryotic cells, sex, multicellularity, social groups. Each of these involved a new level of organization, a jump in hierarchical complexity. So undeniably, the maximum complexity found on Earth has increased over time.
But is it a driven trend? Is there inherent selection pressure favoring complexity across the board?
The consensus is no, it seems to be primarily a passive trend.
Why passive?
Because life had to start somewhere, and that starting point was necessarily simple, the earliest prokaryotic cells. There's a hard lower boundary of simplicity. Evolution can't produce anything less complex than that initial state.
So like the size example, any random exploration of possibilities, any diversification, has to move away from that simple starting point.
Exactly. The variance in complexity can only spread upwards. So while we see these incredible peaks of complexity emerge like us, or complex societies in insects, that doesn't mean there's a universal force pushing all lineages towards complexity.
Because lots of simple organisms are still around and doing just fine.
Absolutely. Bacteria, archaea, many simple eukaryotes, these lineages have persisted for billions of years, often with relatively little change in their fundamental complexity. They are incredibly successful. If complexity were actively and universally selected for a driven trend, you'd expect those simpler forms to be consistently outcompeted and disappear. But they haven't. So the increase in average complexity seems to be more about the expansion of possibilities away from an initial simple floor. And all of this, the role of history, the nature of trends, leads us right into one of the biggest, almost philosophical debates in macroevolution: predictability versus contingency. Was the history of life largely inevitable, bound to follow certain paths? Or was it mostly a series of unique, unrepeatable accidents?
This is the rerun the tape of life idea, isn't it? If we could somehow rewind history and let evolution play out again from an early point, would we end up with something like humans again? Or would it be completely different, unrecognizable?
That's the crux of it. And there's evidence you can marshal for both sides.
Okay, what's the case for predictability? Why might we think evolution follows certain inevitable paths?
Well, first, there are basic physical and chemical constraints. Some solutions just work better than others from an engineering perspective. As the book notes, if you're a large land animal, you're going to need thick leg bones for support, whether you're a dinosaur or an elephant. Optimal design theory suggests certain outcomes are highly probable given certain challenges.
And the really big argument for predictability comes from convergent evolution, right?
Absolutely. We see similar adaptations popping up independently again and again in unrelated lineages that face similar environmental pressures. It's striking.
Like the streamlined body shape for fast swimming.
Perfect example. Evolved independently in fish like sharks, ancient marine reptiles like ichthyosaurs, and mammals like dolphins. Totally different starting points, converging on the same functional shape because hydrodynamics favors it.
Or the classic textbook case of Australian marsupials and placental mammals elsewhere.
Right. You get these amazing parallels. Marsupial moles that look and act like placental moles, the extinct thylacine marsupial wolf, resembling a placental wolf, sugar gliders looking like flying squirrels. Filling similar ecological niches seems to drive evolution towards similar forms, time after time.
So some scientists, like Simon Conway Morris, look at this pervasive convergence and argue, what?
They argue that it suggests the solution space for evolution is actually quite limited. The available pathways are strongly channeled by functional demands. Conway Morris takes it quite far, suggesting that the major outcomes, maybe even including things like intelligence and complex societies, were almost inevitable. The environment basically forces certain outcomes.
Okay, that's the predictability argument. What's the counter-argument? Why believe in contingency, the idea that history is fragile and full of unique, unrepeatable events?
The evidence for contingency is also really compelling. First, look at the major innovations in the history of life, like life itself. As far as we know, it only originated once. The eukaryotic cell, multicellularity, the vertebrate body plan, flowers, feathers. These major breakthroughs each seem to have happened only once. If they were easy or inevitable solutions, you might expect them to have evolved multiple times independently, but they generally didn't.
So their origin depended on a very specific, perhaps unlikely, sequence of prior events.
That's the implication. And then there's the massive role of extinction, especially mass extinctions. These are the ultimate contingent events, unpredictable catastrophes that wipe out entire groups, often regardless of how well adapted they were.
Like the asteroid that wiped out the non-avian dinosaurs.
Exactly. That single event drastically reshaped the biosphere. It cut short the reign of the dinosaurs and opened up ecological space for mammals, which had been around for ages, but were mostly small and marginal to diversify and eventually dominate. If that asteroid had missed, The world today would be unimaginably different. No primates, no us.
They rerouted history completely.
Utterly. And it ensures that evolution doesn't just replace like with like. We don't have equivalents of dinosaurs or trilobites today because their unique evolutionary paths were permanently terminated. Extinction makes history unrepeatable.
This sounds like Stephen Jay Gould's main argument. Rerun the tape, and the tiniest change early on could lead to a completely different outcome later.
Precisely. Gould was a huge proponent of contingency. He argued that if you went back to, say, the Burgess Shale fauna 500 million years ago with all its weird body plans and just randomly let one or two minor lineages survive instead of the ones that actually did, like the ancestor of chordates, then the entire subsequent history of vertebrate life, including humans, simply wouldn't have happened.
Our existence is a fluke of history in that view.
A highly improbable contingent outcome. And this wasn't just Gould. Many major figures in 20th century evolutionary biology, like George Gaylord Simpson and Ernst Mayr, strongly emphasized the contingent nature of evolution, particularly human evolution. They saw the path from an ape-like ancestor to Homo sapiens as anything but predetermined.
So where does that leave us on the idea of evolutionary progress?
Well, if history is highly contingent, and if evolution operates only on immediate advantage without foresight, then the notion of progress towards a specific goal, like humanity, becomes really untenable. Evolution is fundamentally goalless.
It's not climbing a ladder.
No.
Yeah.
It's more like a randomly branching bush, constantly pruned by extinction. The patterns we see, like increasing complexity or trends in size, aren't signs of directed progress towards a predetermined endpoint. They're the cumulative emergent results of microevolutionary forces playing out over immense timescales, shaped by historical accidents, environmental opportunities, and developmental constraints.
It's the outcome of billions of years of local adaptation and chance events, not a march towards perfection.
That captures it perfectly. Tag, outro.
So bringing this all together, what's the big picture takeaway from looking at macroevolution?
I think the main message is that these vast, large-scale patterns we see over millions of years, the origin of mammals, the evolution of complex structures like eyes, the trends in size or complexity, they really do emerge from the same fundamental microevolutionary processes. Selection, drift, mutation, acting generation after generation. There's no need for special macroevolutionary forces.
Simple microevolution writ large, given enough time.
Exactly. And novelty arises mostly from tinkering with existing parts, co-option, and exaptation. Evolutionary rates can vary wildly, with long periods of stasis being common. And critically, there's no inherent direction, no built-in goal or progress driving life up some kind of ladder.
It's a history shaped by stability, punctuated by change, and deeply influenced by the unique, contingent sequence of events on Earth.
That's a great summary. And it leaves us with a final thought to consider.
Good one.
If we accept this strong role of contingency, if we accept that our own emergence as a species was a highly improbable, perhaps unique outcome of this long, winding history, what does that suggest about our own future evolvability?
Meaning, can we adapt to what's coming next?
Right. Given the specific genetic and developmental toolkit we inherited through that unique history, how constrained are we? How readily can our species adapt through natural evolutionary processes to the really novel and rapidly changing environments we're now creating for ourselves? Or does our contingent history limit our future options in ways we can't foresee?
That is definitely something to chew on. A great question to end Unit 4.
Indeed. That wraps up this part of our journey.
And next time, we launch into our final unit. We'll be turning the evolutionary lens directly onto ourselves, focusing on the story of our own species, Homo sapiens.
Looking forward to it. Thanks for joining us for this deep dive into grand patterns of macroevolution.
Thanks for listening to Bookfulness. Join us for the next deep dive.